chapter 11 then is going to be our first introduction to the nervous system. Okay, the nervous system we're going to cover broken into 3 chapters, and we're actually going to leave a little bit of the nervous system off, and then you'll pick that up in A and P2. So we're going to start off kind of giving you an overview. Chapter 11 is an overview of the system, the same way we saw chapter 6 kind of being an overview of the skeletal system. So we're going to start off with a brief description of what exactly is the nervous system, kind of an overview of what happens, talk about the cells that are involved, and then we'll look a little bit at the physiology of those cells. So the nervous system, what you can think about, this is really a control system, right? So this is the master controlling system and communication system of the body. This is also going to be the fast-acting control system of the body. So this is going to be the way the body can pick up information, right? We call that sensory input. So when you have information coming in, the nervous system, that we're going to see there are specialized receptors that take that information in as sensory input, and they're going to bring it into what's called the central nervous system. Okay. And that is an integration. When you integrate something, you take 2 different pieces of information and you put them together. You make associations, you make connections. Okay. So we'll see that there is a part of this that is going to interpret what's going on based on that sensory information, that information coming in. It's going to process it. And then the third component is the response. Okay. Some sort of motor output. We'll talk about an entire system dedicated to motor output. And this could be as simple as skeletal muscle response, which we have already talked about in chapter 9. Okay. So 3 main parts of the nervous system. Now the way this is organized is we're going to break this down into 2 different nervous systems, but please understand these are connected. One cannot function without the other. There's no clear separation between them. Right? So we have the central nervous system. This will be the brain and the spinal cord. This is where we have the majority of our integration. So this is our integration, association, connections, and our command center. So this is where decision making occurs. Okay. The peripheral nervous system, or PNS then, this will be the nervous system of the body. We have different nerves, and we will talk about what exactly is a nerve. Right? And we'll see here we have what are called spinal nerves and cranial nerves. And maybe you can guess that the spinal nerves are going to come off the spinal cord, and the cranial nerves are going to come off the brain. This is what's going to carry messages to and from the central nervous system. So the peripheral nervous system is really the sensory input and the motor output part. Now we can't have one without the other. So the central nervous system needs the peripheral nervous system to bring it information and for it to respond. Right? If you just were a brain in a jar, there would be no way for information to get into the brain, nor for any response to come out of the brain. In the peripheral nervous system, then, there are really 2 main sides of the peripheral nervous system. We have that incoming division, which is sensory. We also call that afferent, right? Afferent with an A. I will overpronounce the following words so that you can really start training yourself the difference between afferent, okay, which is that incoming, and then the motor, or the efferent. Okay. So if you think about the difference between affect and effect, right, in English oftentimes people use those inappropriately and swap them. Right? So think about the difference between affect and effect. Okay. The motor, or the efferent division, okay, with the E at the beginning of it, that is going to be your response division. Now, in terms of sensory information, we tend to automatically, as humans, go to what are called the special senses, right? So I said, what are your senses? Most of us would say sight and smell and taste and hearing. Okay? That's only a very small part of our sensory information. Okay? Those are specialized senses. They're actually very limited in what they bring in. What we have, that 5th sense that we kind of lump into a category called touch, okay, that's actually a much broader category, and that's where the majority of our sensory information actually comes from. Okay, this is where we get impulses that come in, telling us about our external environment, and those impulses come in from our skin, our muscles, our joints, tells our body things about temperature, air movement, humidity, movement of our muscles, how our body is positioned in space. Okay, these are all really important things that we don't classify as our quote-unquote special senses. We call that the sensory afferent fibers, okay, but then we have this other category that's called visceral afferent fibers. Right? Whenever you see viscera, viscera means the organs. Okay? So the visceral afferent fibers, these take impulses from the visceral organs and send them to the brain. So these sense what's going on in your stomach, in your bladder, in your small intestine, in your large intestine. Okay? Now these are not always going to be conscious, but we can think of examples of conscious ones, right? You drink a giant 57-ounce drink at Sheetz, and then you're sitting in lecture, and you suddenly become aware of the stretching and the pressure in your bladder. Okay? Now your body's always aware of how much stretching is going on. You only become consciously aware of it when it becomes a problem. So those visceral afferent fibers, they're constantly monitoring what's going on in your viscera, in your internal organs. And we'll see that in terms of response, there is a kind of important separate part of response system having to do with your organs as well. That the different responses that we can see, we can break these roughly down into two main divisions, and then there is a third one which we'll just mention, but we won't really talk about. You will pick that up in A and P2. The first is the conscious control of skeletal muscles, and we've already talked about this in Chapter 9 when we looked at how skeletal muscles receive impulses. Now we're going to talk about the neurons behind it. Okay, we talked about what happens in the muscle, now we're going to talk about the neurons. So the somatic nervous system, whenever you hear somatic, somatic means the body system. This is where we have the conscious control of skeletal muscles. This allows for our conscious movement. It will also allow for some programmed movement. And we're going to see that it's going to interact with different parts of the brain, which we'll learn in Chapter 12. So the idea of walking. Walking is a programmed movement or a complex movement, but most of us don't have to think about everything you're doing as you walk. And we'll see what that is the somatic nervous system interacting with different parts of the brain that give us these programmed or repeated or patterned behaviors. The second part then is the autonomic nervous system, or the ANS. Okay. This is, you can think of as the autonomic, right? Think automatic. This is the involuntary portion of your response. This is where you're going to have regulation of your smooth muscle, right? Lining your hollow organs, smooth muscle, in terms of your stomach, how much your stomach is contracting, your small intestines, your bladder. Okay? Most of that you don't have conscious control over. Also the cardiac muscle. Now the cardiac muscle, we did say the cardiac muscle, the beating of the heart is intrinsic to the heart, but there are ways that your nervous system can speed up your heart or slow down your heart. Also then the glands, and we briefly talked about glands in Chapter 4 when we looked at epithelial tissues. The glands are going to be very important in what's called the endocrine system. Right? The endocrine system is another control system of your body. In fact, the endocrine system and the nervous system often work together. The endocrine system leads to more slower and long-term responses, where the nervous system is faster and shorter-lived responses. So the autonomic nervous system, then, we have the two divisions, the sympathetic and the parasympathetic. The sympathetic you've heard of before, called fight or flight. The fight or flight system, this is where you're going to have these short bursts of high stress, and you'll see changes, things like getting your body ready to run away or fight. So your heart starts racing, your breathing starts increasing, your muscles tense up, blood gets diverted away from your digestive organs, right? This is why if you are ever driving on the highway and you nearly lose control of your car, a truck runs you off the road, afterwards you have that feeling where you can still feel your heart racing, you feel out of breath, you sometimes feel nauseated, where you just feel off. That's your body coming down from a sympathetic rush. Okay? That is going to be due to what the layperson would consider adrenaline. Adrenaline really is epinephrine and norepinephrine. They're two different hormones released from two different parts of the body. Okay. The other system is the parasympathetic system, and the parasympathetic system is the rest and digest system. So the parasympathetic system is what hopefully all of you are in right now. Okay. Hopefully you're not all under high stress. The rest and digest system is where the body is trying to store excess energy, where the body is trying to focus on things like the reproductive system, building new cells. Okay. And we won't go too much into the sympathetic versus the parasympathetic nervous system in A&P 1, but you will see it in A&P 2. So we will pick up now talking about the histology of nerve tissue. Okay. So within the nervous system, we're going to see there are two main types of cells. Okay. The workhorses, the ones that actually do the job of the nervous system, which is transmitting signals, sending signals, communication, those are called neurons. These cells are excitable, so they're like muscle cells in the sense that they can transmit electrical impulses, which should tell you that we are going to have to talk about action potentials. And then there are also an assortment of different supporting cells. And these are going to be cells that are going to support neurons, whether it be physically support them or chemically support them. And we're going to actually start off with these supporting cells. And we're going to give an overview of these supporting cells and talk about a few examples, and then we're going to get into the neurons and what they do and how they do it. So overall, we can call these supporting cells neuroglia or glial cells. And there are a few different jobs these can do. Sometimes, as I said, they can provide a physical support, so they can provide a scaffolding to actually have the neurons be in the right place. We will see a very important function is that they help to segregate and insulate neurons. Right? We're talking about electrical signals here. Right? One of the rules of electricity is you need to have insulation on your wires. Well, you can imagine neurons are like little itty-bitty tiny electric wires. These neuroglia may also help to make connections between neurons and guide neurons, as well as supporting them physiologically in terms of health and growth. So the first kind of cell that we will talk about for these neuroglia cells are called astrocytes. Okay. Astro, when you see astro in a word, maybe you think about astrology or astronomy. Astro means star or star-shaped. These have a rough starburst pattern, star-shaped. See how they have lots of branches coming out? Okay. These astrocytes, these are the most abundant and versatile. Right? You see the most different jobs here. These have a high degree of branching, hence that they look kind of like a starburst. One thing that they can do is they cling to neurons. They actually wrap around neurons. You can see they have these extensions coming off. They can also wrap around capillaries, and in this way they help keep neurons and capillaries close together. Right? That's going to allow for these neurons to get nutrition, to get rid of wastes, things like that. They also help to physically support and brace neurons. They're anchoring these neurons in place. The other thing that's going to be very important is they help to guide the migration of young neurons. Now, we're not going to talk about the development too much, but neurons are very interesting in the fact that most of the neurons that you have during embryonic development are the neurons that you're going to have for the rest of your life. The neurons do move, they relocate in order to develop the entire nervous system. So that movement or migration is very important. They also, because they can attach to both the neuron and to the blood vessels, they're going to help to control or monitor the chemical environment. So those are the astrocytes. The other cells are called microglia. Right? These are much smaller. These have an oval shape, and they have processes that come off. Right? The word process, when you talk about a process on a cell, a process is a branch that comes off. So this is a process. So here we see this has many processes. These cells, these microglial cells, can be phagocytic, so they can actually engulf and break down debris, dead cells, dying cells. These help to monitor the health of neurons, so to tell if a neuron is chemically healthy or physiologically healthy. A very important type of these neuroglia are called ependymal cells. Ependymal cells look a lot like epithelial cells. These have varying shapes. Okay. These usually have two different sides to them. So they have a side that's going to sit. They have a side that is going to sit on the brain or spinal cord tissue. Okay. And they also have a side that's going to face out into a fluid-filled cavity. We will see that the brain and spinal cord have within them a hollow opening that runs through that we call ventricles and a central canal, and that is full of a fluid called cerebrospinal fluid. That fluid is going to be very important to the health of the central nervous system, and these ependymal cells will help to monitor and create that cerebrospinal fluid. Okay, so those are the ependymal cells. The next kind of cell is called an oligodendrocyte, and people always freak out when they see this name. It's really not that hard. It's oligodendrocyte, right? When you see this, this is a cell that you would find specifically in the central nervous system. And what this cell does, it's going to create something called a myelin sheath. Myelin sheath is basically insulation for neurons. It prevents the axons of two neurons from touching one another and basically shorting each other out. It's also going to help neurons when we look at more in depth how action potential occurs. We're going to see it's going to help action potential to occur, and it's going to help to speed up action potential. Now, in the central nervous system, the neurons of the central nervous system are all really, really close together, and they're all pretty small. So what you see is you may see a single oligodendrocyte, and it has a process that comes off, and it creates a flattened piece that actually wraps around the axon of a neuron. It may have multiple processes that come off and wrap around different neurons. So one oligodendrocyte may wrap its hands, if you will, around a few different neurons, creating this myelin sheath. We only see these in the central nervous system. In the peripheral nervous system, the neurons are going to be much longer. They have to travel much bigger distances. They're more separate from each other. They're not as tightly packed together. So here we see a different kind of cell that does the same job. Okay? These are called Schwann cells. Okay? These are also called neurolemmacytes, because we're going to see they're going to create what we call the neurolemma. So neurolemmacytes make the neurolemma. These will surround the axons of cells in the peripheral nervous system. Okay? So here is a cell. This is a typical, what we would call a unipolar cell. Okay? This would be what's called its axon, and that axon is the conducting region. And all along here we have these little areas. Each of these is an individual Schwann cell. That Schwann cell has been flattened out, and it wraps around the axon, and it creates insulation. There's another kind of cell that might also do that, and these are called satellite cells, and the satellite cells wrap around the cell body rather than the axon. So make sure you keep straight that the myelin sheath is created by oligodendrocytes in the central nervous system and Schwann and satellite cells in the peripheral nervous system. Now, since we're talking about myelin sheath, this is a good time to take a look at it and see what it is. It's a little bit important here. Myelin sheath is basically created of mostly proteins and lipids. It's found around most of the long axons of neurons. There are some neurons that are unmyelinated. If you have a very, very short neuron, we'll see that it doesn't necessarily need to have myelin. It's going to help protect the axon, insulate the axon, and increase the speed of nervous impulse transmission. Now, the fact that it's made of proteins and lipids, okay, and I'm telling you it's created by cells, that should hopefully get you thinking about what part of the cell is making the myelin sheath, because it's actually quite interesting. The way this is made, if we use the example of a Schwann cell, the Schwann cell is a cell like any other cell, so it has a plasma membrane. That plasma membrane is phospholipids with proteins in it. Okay? That plasma membrane then, if you take that Schwann cell and you imagine almost rolling it out flat like pizza dough and then wrapping it around an axon, what you get is you get multiple layers of plasma membrane with very, very little cytoplasm in between. Okay? And that's basically what happens here. Here is the Schwann cell. It's a cell, it has a nucleus, right? As this wraps around the axon, we see that most of the cytoplasm gets pushed into the very outer layer, which we will call the neurolemma. And then in this region here, the actual part that's considered the myelin sheath, you basically have plasma membrane, plasma membrane, plasma membrane, kind of all squished together. So that's going to create that protein lipid layer. It's just the plasma membrane of that Schwann cell. So when we talk about these being neurolemmacytes, it's because when they make the myelin sheath, that last layer we call a neurolemma. So these are the cells that make it, the neurolemmacytes. In the nervous system, in the peripheral nervous system, these individual Schwann cells, they don't overlap. They leave very, very tiny gaps in between each successive piece of myelin sheath, and these are going to be very important. Okay, we call these nodes of Ranvier, or neurofibril nodes. These are going to be extremely important in how action potential propagates or moves along an axon. These are also going to be sites where we may see an axon branching, okay, into what are called axon collaterals. So keep that in mind, because we will take a look at that. Any questions on the glial cells? All right, so you should be able to list them and give the basics of what they do, as well as recognizing the basics of the shapes. Did I say it was? Okay, good. So let's just finish up a quick overview then of the neurons themselves. We're going to look at the anatomy of the neurons. We have to understand the anatomy of the neurons before we can talk about the physiology. The neurons that we're going to talk about all have the same basic parts, but we will see them organized slightly differently. Okay, so they're all going to have a body, which we may also see called a soma. They're going to have an axon, which I've been talking about already, and they're going to have dendrites. Now, neurons are really cool in the sense that they are long-lived, amitotic, and they have a high metabolic rate. So what does that mean? Long-lived. Neurons that you have now are neurons that you had since you were an embryo. Okay? Hopefully, your neurons aren't going to die because they are amitotic. If you remember, mitosis is the process of how one cell divides to become two new cells. That's how cells reproduce. Neurons don't do that. Neurons do not replicate, they do not regenerate. Okay. What that means is if you kill your brain cells, they're gone. They don't come back. Okay. Neurons also have an extremely high metabolic rate. They are always active. Okay, so they always need oxygen. They always need to get rid of waste. They always need a source of making ATP, which is usually glucose, though the brain can actually use other things as well, things called ketone bodies and a few other things. Okay. This is why if you have somebody who drowns or suffocates and they're able to bring them back in terms of getting their heart beating again and getting their lungs working again, they sometimes have what's called brain death. right, or severe brain damage. The cells in the brain will die first. If deprived of oxygen, your brain cells are the first to go. Your body cells may be able to come back, but because the brain cells, they use up so much energy so quickly, right, they are the first to starve, and they are the first to die off. Now, one of the reasons for this is because they are constantly sending signals, right? Your nervous system is constantly working, constantly. The big thing that it does is electrical signaling, right? So we're going to talk about the plasma membrane, we're going to talk about electrical signaling, we'll talk about those action potentials, and we're going to talk about a new kind of signaling called a graded potential. Now, we don't get into development too much, but the way neurons develop really has a lot to do with early on cell-to-cell signaling, right? How the neurons are told to grow and change connections. So let's give a quick overview of the parts of the neuron. This is a typical type of neuron. This is called a multipolar motor neuron. This is the neuron that we will use as a model, right? Just like when we first looked at the cells, we looked at a general cell. This is our general neuron. Okay? There are different kinds of neurons, and we're going to look at different shapes of neurons, different structures of neurons, but here's our basic neuron. Okay? It has a cell body, and that cell body has all of the parts that make cells cells, right? So it has a nucleus, it has a nucleolus, it has rough endoplasmic reticulum, which have a special name in neurons called Nissl bodies. This is the cell body here. You can see it looks like a cell body. What's different about it, it has stuff coming off of it. And those projections, those are going to be what I will refer to as processes. Okay, a process. Now there is a very special area here at the base of the cell body, right, where the cell body and the axon kind of connect. It's a cone-shaped region. It's called the axon hillock. Okay. That axon hillock is going to be super important in terms of understanding where the action potential kicks off. Now, off of this cell body, we may have these branches called dendrites, and we'll see this is one of the things that varies greatly among different neurons. So some are going to be multipolar as this, multipolar meaning they have many projections coming off. This has many dendrites. These dendrites branch. These are the receptive regions. So this is where the neurons are able to pick up the signal coming from the previous guy in the line, whether it be another neuron sending them a signal or some sort of sensory cell sending them a signal. Okay. This is the receptive region where the signal from the previous will be picked up. Okay. This is where we're going to talk about that new thing called graded potentials. Okay. So what happens here is called a graded potential, and we'll see how it's different than an action potential. Then down over here we have this long process that comes off. Sometimes you see branches on the long process, but for the most part they're usually a singular process. Okay. That is the axon. The axon is what is going to send the signal. Once it gets received by the dendrites, processed by the cell body, it will be conducted by the axon down to the axon terminals. Right? And this is the secretory component. This is where that electrical signal is going to get sent to the next in line. And we've seen one type of this already, right, when we looked at neuromuscular junction. The neuromuscular junction was where this axon terminal talked to muscle. So that's kind of the end game of things. So now that we've looked at the main structures of the neuron, we're going to talk a lot about what goes on in the axon. Okay, the axon is going to be where we will see the generation and transmission of action potentials, which is really the signals that are going to be important in the nervous system. We've already seen a sneak peek of this, especially what goes on at the axonal terminals or the axon terminals, which is where we're going to see the secretion of neurotransmitters. And in the skeletal muscle system at the neuromuscular junction, we saw one example, which was acetylcholine. Now, two important terms that you want to become familiar with. In the movement of signal along the axon, the way the movement goes in most axons, the way it's supposed to go for the most part, is what's called anterograde. Okay. In this case, and we're going to see why this is and the way the action potential occurs, we really only want signals moving one way. Right? come in from the dendrite, process in the cell body, pass from the cell body into the axon, axon down to the axon terminals, next neuron. Okay, it's like a one-way road. Okay. We call that anterograde transmission. Okay, so we have movement of the signal in the forward direction, right? The anterior direction, right? Anterior is towards the front. There are some cases where retrograde transmission occurs, not usually in humans, but retrograde is movement away from the axon terminal. Okay. So just to put those terms out there, we're going to think about things in the one way, right? One direction. Dendrite, cell body, axon, axon terminals. Another important terminology that you need to clarify here, when we talk about the brain and spinal cord, we're going to differentiate between what we call gray matter and white matter. Now, you may have heard somebody refer to the brain as your gray matter. Okay? There is both gray and white matter in the brain. White matter is where you have collections of myelinated fibers or axons. So where you have lots of axons and those axons have lots of myelin on them, you have a high amount of those lipoproteins, the fat protein complexes, so it actually looks white. Okay? If you think about what fat looks like, if you look at like a cut of meat, it tends to have a white appearance to it. Now the gray matter is where the cell bodies are going to be, right? And where the cell bodies are, you can imagine that's also going to be where the axon terminals of the previous neuron are going to be. So that's where we the actual connections going on, right? The synapses. We will also see that in the gray matter, sometimes we have unmyelinated fibers. So if you have very, very short axons, they don't necessarily need to be myelinated. Now, this difference between gray and white matter will be extremely important when we look at the brain, we look at the spinal cord. So you want to kind of start thinking about that right away. You don't see gray and white matter in a single neuron per se, but depending on how those neurons are arranged, if all the cell bodies line up, you see gray matter. If all of the axons are lined up, you see white matter. Yeah. So in terms of the different kinds of neurons we might see, the first example that we looked at, I said, was called a multipolar neuron. This is a neuron that has 3 or more processes coming off. So usually one axon with many dendrites. Okay. This is the one we'll look at in lab. Right? This is our classic motor neuron, which is what we'll talk the most about. The other kind are called bipolar neurons. Bipolar means there are 2 main processes coming off, 1 axon and 1 dendrite. Now that axon and the dendrite might branch at the end, so you may see axonal terminals and you may see dendritic branching. Okay? But coming off the cell body, there's just 2 main branches, 2 main processes. The last one is what's called unipolar or pseudo-unipolar. This is where you have a single branch or a single process coming off the cell body, and then that process spreads out, and you may then have a very long axon off of that. So let's look at some of these examples. This is what we have seen previously. Okay. Here we have this multipolar, right? Many branches, many processes coming off. Okay. And when we looked at this, I mentioned where you have a receptive region. Now the receptive region here is shown in this kind of blue color. Where the dendrites are is the receptive region. Dendrites are going to receive the incoming stimulus. Then we have a single long axon. In this case, it is myelinated. That myelinated axon, this is the conducting region. This is where the signal gets conducted. That signal is going to be in the form of an action potential. And then finally, at the end here, we have this branching axonal terminal. This is where the next signal gets sent on by the secretion of neurotransmitters. So this is the secretory region. This is the type of neuron we would see when we look at a motor neuron. Here we have an example of a bipolar neuron. Notice the cell body has 1 process, 2 process. Okay. The dendrite then is a very long extended dendrite, so we have a much longer receptive zone or receptive region. The axon would be our conducting region as seen before. Notice this one is not myelinated. And then finally we have our secretory region, where we have the axonal terminals. This is very, very common in terms of what you would see in some of the special senses. This is what the cells of the retina look like. Some of these cells do not actually create action potentials. They use just what we're going to talk about as graded potentials. Graded potentials work really well over very short distances. Okay, and we're going to talk about what those are. So some of these bipolar cells actually don't use action potentials. They use this different form of electrical signal called a graded potential. Now this unipolar or pseudo-unipolar, here we have a cell body with a single process coming off. Okay. We still do have receptive endings. The receptive endings, though, tend to be much smaller, so we see a much smaller proportion of this cell being dedicated to reception. This is all considered the axon, so this is all where we would have the conduction of the signal. And then finally the secretory region where the signal gets sent on. These unipolar or pseudo-unipolar cells, these are very typical for our sensory cells. Okay, so the cells are going to bring information into the brain and spinal cord, and these are going to be really, really long neurons. Okay. Same with the motor neurons, because you can imagine there are neurons that have to go from your spinal cord to the tip of your finger, okay, both in and out. So we're going to see there are single cells that can be meter long. Some of the examples, we won't look at all of these, but just some of the examples and some of the different ways you can see multipolar, bipolar, and unipolar. These are both cells that you would see within the brain. We will look at one of these. This is called a pyramidal cell, and this is called a Purkinje cell. These cells are going to be very important in the brain and in the functioning of the brain, and they have a very distinct look to them, a very distinct shape to them. Classic multipolar. These are bipolar cells. This is an olfactory cell, and this is a retinal cell. Okay. Notice that these are both involved in those special senses. We don't have lots and lots of them, right? But for the most part, these are going to be involved in special senses. And then here is our example of a pseudo-unipolar cell, and as I said, this would be found in what's called the dorsal root ganglia cells, which are a special kind of sensory cell. In addition to classifying these neurons based on what they look like, we can classify them based on what they do. And we've already alluded to what they do a little bit, okay, in terms of the fact that we have the sensory and the motor parts of the nervous system, the afferent and the efferent, okay, parts of the nervous system. There's also association neurons, right? So the sensory neurons, these are going to be neurons that you would find bringing information into the central nervous system, right? They're sensing things. The motor neurons are going to be part of that motor output, okay? So they're going to be taking impulses away from the central nervous system. And then these interneurons are going to be really important. These are association neurons, and these will send signals within the central nervous system. Okay, when we look at the brain, when we look at the spinal cord, we're going to see there are a lot of connections in the brain. So a signal comes into one area of the brain, and then it has to go from that area to another, from another to another, in order for that signal to be sent around and properly processed. And those neurons are all going to be called interneurons. So we can kind of superimpose this idea of the structural classification and the functional classification and look at some examples. This right here is something we will be talking about, and if you imagine taking this diagram and superimposing this diagram on it, we will see that this is what's called a reflex arc. Okay. Here we have two examples of multipolar neurons. Okay, most interneurons or association neurons that are found wholly within the central nervous system are multipolar neurons. Okay. Motor neurons are multipolar neurons. Right? So these are two examples of multipolar neurons. One is an interneuron functionally, the other is a motor neuron functionally. As I said, we have these special examples of bipolar neurons. We're really only going to see those in the special senses. The other one that's going to be very important, here's our pseudo-unipolar neuron. This is going to be classic sensory neuron. And then a very important thing we will look at when we look at the nerves. Notice over here, outside of the spinal cord, that's where the cell body is going to be. Right? All of the cell bodies are going to be in the same place for these sensory neurons, in what's called a dorsal root ganglia. And that will be a very important structure when we look at the spinal cord. Right. So we're going to jump then into talking about the neurophysiology. So just some quick terminology. We've seen this before. When we talk about cells that are irritable, irritable means they're excitable. Okay? When we have something that's excitable, it means that we're going to see it has the ability to change something about its membrane potential in order to send electrical impulses. And those electrical impulses are really going to be due to changes in ion concentration and ions moving across the plasma membrane. Right? So we've seen a sneak peek of it in Chapter 9. Now we're going to really kind of get into the nitty-gritty of it. So to review some of the information we started looking at in Chapter 9 having to do with action potentials. Right? I want to clarify the ion channels. Right? If you think back to how any cell is organized, there are plasma membranes. Okay? Plasma membranes are phospholipids, so they do not allow things that have charge to pass. Ions, by definition, have charge. They're either plus ions or minus ions. So they cannot pass through a plasma membrane unaided, so they have to have some sort of channel to pass through. Right? We looked at this in Chapter 3, and those channels are going to be proteins in nature. Now we have some channels that are passive channels. Right? These channels are always open, right? So they're not gated. These are always open, so ions can always leak out of or leak into the cell. So you will also hear these referred to as leaky channels. And with these passive channels, the ions will move along their concentration gradient. Okay. And we will see that these leaky channels, or these passive channels, are going to be important in establishing the resting membrane potential. Of course, we are going to have some channels that are gated, right? And by gated, what I mean is they are closed under some situations and open under others. And when they're closed, ions can't pass through. When they're open, ions can pass through. So the gating mechanism then is going to be what changes between these, what's different between these. Right? They are all going to be specific for either one or two ions. We can have chemically gated channels. These are also called ligand-gated channels. These are like a lock and a key. So we actually have to have binding of a chemical, usually a neurotransmitter, to the protein. When that binding occurs, it changes the shape of the protein and opens the channel. Okay, so those are chemically gated. The voltage-gated channels, then, these are going to open and close in response to a membrane potential. And what we will see with these are more like a door that has a lock on it that you just wave a card in front of, these electrical scanny doors. These are channels that at resting membrane potential are closed, and depending on the change in voltage, the change in the membrane potential, there's a certain key or threshold voltage that's going to change the shape of the protein and open that gate. Those are the two big ones that we will focus on. There is another kind that is very important for our sensory system, specifically the sense of touch, and these are called mechanically gated channels. Mechanically gated channels, these channels open and close in response to being pulled and pushed open. Okay. These are going to be especially important in our sense of touch. So when we have things like those Meissner's corpuscles, those Pacinian corpuscles, okay, those Merkel discs that we saw way back in Chapter 5, those are all things that have mechanically gated ion channels. Okay, so when they're pressed and actually squished, it opens channels and it's going to kick off electrical signals. Right? We won't focus on those too much for now. Now you can take a look at this visually. When we have a chemically gated ion channel, again, lock and key. Now what is the chemical? Well, depends on the channel, right? So we have a neurotransmitter. There are a lot of different neurotransmitters. Okay, we've talked about acetylcholine. Okay, we're not going to go through all of the neurotransmitters. Okay, but neurotransmitters are very diverse, and there's a lot of different things that can act as neurotransmitters. Right? This particular channel is a is a channel that is so not selective, so it allows either sodium or potassium to pass through. Okay, some channels are much more selective; they only allow one or the other to pass through. Our voltage-gated channel, these will be very important in how action potential works. Okay, so at resting membrane potential, where we have a relative negative inside state, these are closed, and then when we change that potential, right, change that resting membrane potential, it opens. And again, these purple blobs, these are just representing the proteins. The proteins have some complex three-dimensional shape. Okay. Now, if you open these channels, okay, the channel itself does not dictate the direction that the ion is going to move. Okay. What dictates the direction that the ion is going to move has to do with the concentration gradient of that ion. Right. It also has to do with the electric gradient. If you want to think about this as the charge gradient. So you're not only going to see, say, sodium ion moving from where there's more sodium ion to where there's less sodium ion, but it's also going to be affected by the relative number of charges, okay, because sodium is positive. Positives, lots of positives together, should repel one another. Whereas where there's less positives or possibly more negatives, the positives should be attracted to that as well. So the movement is really more complex than just where's there more sodium, where's there less sodium. Okay. So what we have is a different kind of gradient where we have what's called an electrochemical gradient. The electrochemical gradient is a combination of the gradients of the charge and the concentration. Now, when you have the movement of these ions, that's what's going to create your electrical current. Okay. Now, I'm not going to get into the physics of electricity because it makes my head want to explode. Okay. But in a simple sense, electricity is movement of charged particles. How many particles are moving across a certain area is measured as voltage. Okay. So that's really what we're talking about here. We're talking about how those charged particles are moving, who's moving more, what's moving in what direction, and overall that difference is going to be measured as voltage. Okay. So you can't really think of resting membrane potential and action potential and gradient potential as being static, as being a set number. It's a relative movement that gives us that set number. And I previously explained the electrochemical gradient part. Right? Make sure that you are comfortable with that. Previously, when we discussed diffusion, right, we said that things diffuse down their chemical gradient, so they go from areas of high concentration to low concentration. I feel like we're all comfortable with that. Now we have to add in this idea of the electrical gradient, where ions are going to move towards the opposite charge. Right? If you think about that, that makes sense. If you have two pluses, they're going to repel one another, but a plus is attracted to a minus, right? Opposites attract. So those two things taken together are really going to dictate how ions move when channels are open. Right? When channels are closed, they can't move. That's what builds the potential. Now, that being said, I said that this resting membrane potential isn't static. It's really movement. There's relative movement. In order for us to look at this on a picture, we're going to have to try and get a little imaginative. Right? We will see an animation in the mastering A and P. There is an animation called Establishing Resting Membrane Potential, which helps to clarify this, and we will see that. Okay. So, when we looked at muscles, we had talked about resting membrane potential. Right? In neurons, it's the same basic thing. We have a potential difference if we were to measure right across the plasma membrane. We see a potential difference we measured at minus 70 millivolts. Again, that's an approximate number. We're going to give you one set number to think about, but there are ranges. Okay. What that means is across this membrane, based on the movement of potassium and the movement of sodium, there is a difference that the inside is relatively less positive, therefore appears negative. Now, the reason that we see this has to do with this kind of close-up over here, where you can actually see our plasma membrane. So this is our phospholipid bilayer. Right? Here is a sodium-potassium pump. That was our example of active transport. That's going to actively pump sodium out of the cell and pump potassium in. Now the key thing to look at here as well are these arrows showing you potassium diffusion and sodium diffusion. Now, please, these arrows are a little misleading in the sense it looks like the potassium and the sodium are just diffusing right through the plasma membrane. That cannot happen. These are representing the overall net result of those passive channels or those leaky channels. Okay? So there's some amount of channels that are always open, and they're always allowing some amount of potassium to leak out and some amount of sodium to leak in. But the sodium-potassium pump is kind of working against that and is always pumping sodium out and pulling potassium in. So it's working against its own diffusion, and that's actually creating the concentration gradient that makes sodium want to move in. Right? If you have more sodium outside than inside, it's going to want to move in. If you have more potassium inside than outside, it's going to want to move out. Now, there aren't tons of these leaky channels. Overall, there's more leaky channels for potassium than there are for sodium. What that establishes is this resting state. Now, you can actually take a look at the relative concentrations. Right? So for sodium, at resting membrane potential, we have approximately 150 millimolar concentration on the outside and 15 millimolar concentration on the inside. Right? Millimolar is a concentration. You don't really have to worry about what it means. You will hear it in the future. Okay? It's a way of measuring numbers of particles. But what this basically means is there's 10 times more sodium outside than there is inside. Right? Now for potassium, there's about 150 millimolar concentration of potassium inside, 5 millimolar outside, so it's even higher. It's like 30 times more inside than outside. Okay. Then we have some other things. So far, we've only ever talked about positive ions, right? But you can't have only positive ions. There have to be negative ions. This cell is not positive. Okay. It doesn't have an overall positive charge. This cell is not negative. It doesn't have an overall negative charge. Okay. That minus 70 millivolts is a relative difference just across the plasma membrane. So we have to have some anions, some negative ions here. One of the big ones is going to be the chloride ion. Okay. The chloride ion has a different, there's a potential here as well, but we're not going to talk really about movement of the chloride ion. Right? If you can imagine that there aren't a lot of channels for it, then its movement becomes not very important. It kind of just stays put. The other thing then is this A minus. This is a general anion or protein anion. It's lots of other things that we kind of lump together to look at a single number that all are negative. Okay. So this is what's going on within a neuron. Okay. This is where we're going to start off. In order to look at how action potential, gradient potential, these electrical signals work, you have to understand resting potential, and you have to know it. So, more sodium outside than inside, more potassium inside than outside. Okay. Overall difference, minus 70 millivolts. Okay. Meaning that there's more positive charge lined up on the outer surface of the membrane than on the inner surface of the membrane. So if we can change this, that's going to be a signal. Right. This is where that signaling is going to come into place. And in order to change that resting membrane potential, really what we have to do is change the cell's permeability or the membrane's permeability to ions. And what we mean by that is opening channels. Right. So if we open channels that are gated, we can change the way the ions are moving. We can change that resting membrane potential, and that's how we're going to create our action potential and also those graded potentials. So we're not adding ions into the system. We're not taking ions away. We're just moving them. We're shuffling them around by opening and closing channels, and that's going to give us our signal. And that signal is going to be what allows the nervous system to work. Question? So in terms of how we can change this membrane potential, there are three key ways or three key events that are going to change membrane potential. The first is what's called depolarization. Right? And in depolarization, the inside of the membrane becomes less negative compared to the outside of the membrane. Right? What that means is the inside and the outside are becoming more alike in charge. Right? And then eventually we may see a reversal of membrane polarity so that the inside becomes positive relative to the outside. That's going to be due to movement of ions, right? So we're going to be, you can basically imagine it as like filling up holes and then digging holes out. Now, repolarization, therefore, is the opposite of depolarization. This is where the membrane is returning back to its resting state, so the inside is becoming more negative again relative to the outside. There is a key thing that happens in neurons. It's called hyperpolarization. Hyperpolarization means that the inside of the membrane becomes even more negative than it was previously. Now, these are relative terms. So if you can imagine rest is minus 70 millivolts, that means there is a distance of 70 between the inside and the outside. We're not talking about a length here. We're talking about a charge difference here. Okay. Depolarization, those two things become closer and closer together, right? So that minus 70 becomes smaller, 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 and then we may see that reversal. The repolarization then is as those move back to becoming more different again. Hyperpolarization then actually expands that different, makes things even more different. So we're going to see the numbers going into the minus 80s, minus 85s. Okay, so we have even a bigger difference between the inside and the outside. That hyperpolarization is going to be extremely important in how we have only anterograde transmission. How do we keep signals moving in just one direction? Now, as I said previously, there are ways to actually measure membrane potential. Okay. We have little tiny voltmeters that you can actually touch to a cell and measure it. So let's use that in our imagination. We're not going to do it at all, right? But we'll use this in our imagination. Let's imagine that we can measure what's going on in terms of the membrane potential of a cell, right? When a stimulus comes in, when a signal comes in, it can actually have two different effects. The first effect is to cause a depolarization, right? We call that a depolarizing stimulus. Okay. When you have a depolarizing stimulus, we go from resting membrane potential of minus 70, and we see a slight depolarization. Now, this is not an action potential, right? We're backtracking. I'm going to tell you how that action potential comes to be. Okay. This depolarization, we're going to see the way that it works is if there's enough of these, or if this happens frequently, what may happen is that's going to kick off the trigger to make an action potential. Okay. This is how we try to go through and excite cells. Right? We're bringing them closer to an action potential. Okay. So we can call this an EPSP, or an excitatory postsynaptic potential. Whenever you talk about a synapse is where you have two neurons meeting. Okay. You have a presynaptic neuron, that's the person sending the signal. Then you have the synapse, which is a space. Then you have the postsynaptic neuron. That's who's receiving the signal. Right? So here we're looking at what's going on in a neuron that's receiving a signal. Okay? If the signal coming in or the stimulus coming in is leading to some depolarization, it is exciting the neuron. It's trying to get that neuron to send a signal. On this trace, we have a hyperpolarizing stimulus. This is taking, rather than depolarizing, it's hyperpolarizing the cell. It's making the inside and the outside more different. It's bringing it away from making an action potential. It's in some ways blocking an action potential from happening. So this is called an inhibitory postsynaptic potential, an IPSP. So this is going to be the way we'll see inhibition. Now, we won't talk a lot about inhibition. We're going to really focus on excitatory cells or excitatory postsynaptic potentials. But I wanted you to see that there are both. Right? And that's a really important component of how the nervous system works. Now, I had alluded to these new things called graded potentials. Okay. So far, we had pretty much looked at action potentials in the way that muscle cells work. In neurons, there's a very important component called graded potentials. Now, graded potentials are what we're going to see occurring in the dendrites. This is going to be where we have short-lived local changes in membrane potential. This is usually going to be due to the binding of a ligand or a neurotransmitter to a chemically gated or ligand-gated channel. These decrease intensity with distance, right? So if you can imagine where your initial stimulus is, the response will be greatest right at that stimulus point, and the further away you go, the weaker and weaker the signal gets. Right? The other thing that's key here is the magnitude of this response changes with the stimulus. So if you have a stronger stimulus, you have a greater initial response, which could then go a little bit further. Right? These graded potentials are great in a very short distance. Okay? These cannot send signals for very far distances, so we're going to see these graded potentials are really only going to be used in taking signals from the dendrites and bringing them into the cell body, or in some very, very short neurons. Okay? Now, if you want to try and imagine what a graded potential is, right? The way I visualize graded potentials as opposed to action potentials, right? If you imagine a still pond, okay? Imagine a still pond, and you take a rock and you throw a rock in the center of the still pond, right? You're going to see waves that come out, and those waves are going to be bigger right where the rock lands, and the further away from where that rock lands, the waves get smaller, and then they go away. And at the edges of the pond, you don't even see any response, okay? That's basically what a graded potential is, right? Now, depending on the size of the rock, if we take a boulder or a much bigger rock, we throw that in, we're going to get much bigger waves initially, and those waves are going to go further, right? They're going to be able to go out further. As you're trying to imagine what's happening in a neuron, we're not throwing rocks to the neuron, right? Instead, what we have is we have chemicals binding to channels. When that chemical binds the channel and opens it, some number of ions are going to come in. And as those ions are going to come in, they're going to create a wave of depolarization. But that's going to be focused. The stimulus point is going to be, where was that chemical released? How much chemical was released, right? If a lot more neurotransmitters released, it can affect a lot more channels. We'll get a bigger initial response. Now, there's two important things that come out of these graded potentials as well. These things can add up. Okay? We call it summation. And this summation is going to be extremely important in how signals that come into the cell actually turn into an action potential. Because we can't have every single signal that comes into the cell, we can't have it leading to an action potential, because we basically burn out our nervous system. So we have this thing called summation. Right? There's two main kinds of summation. One is called temporal summation, and one is called spatial summation. And temporal summation is if you have a stimulus that's repeated and repeated and repeated and repeated and repeated, the response gets progressively bigger, right? Until it's big enough that it leads to an action potential. Right? So thinking about our same pond again, you take a rock, you throw it, as soon as it hits the water, you throw another, and another, and another, and you keep throwing a rock, keep throwing a rock. Eventually, those waves start to build up on each other, and you get much bigger waves that can go further. And if they go far enough, and hit the shore, they're going to kick off an action potential. Okay? So we have one rock, throw it in, get some waves to go away. Same rock, we do it over and over and over. Those waves get bigger and bigger, and they go further and further until they're big enough that they hit the shore. That is temporal summation, or summation over time, right? Repeated exposures. The other one is spatial summation. Spatial summation is when you have lots of stimuli throughout the cell, all sending signals in at the same time. So instead of having your single rock that you throw in and the waves go and then they... Right. Now, every single one of us has a rock, right, all the same size, and we all throw those rocks into the center at the same time. So now we have 20 rocks hitting at the same time. That creates much bigger waves, which can spread out and hit the shore and lead to an action potential. So those graded potentials are very different than action potentials. Okay? Graded potentials are great for limiting what kinds of signals lead to action potentials, but these graded potentials are not good over large distance, right? Because the further they go, the weaker they are. So we need something very different in order to send signals far distances. And in the body, we really are talking about some pretty far distances, because we have some neurons that have to go from the cortex of our brain all the way to the tip of our finger, right? And vice versa. Now we can look at this graphically as well. Now this is a very different scan than we've seen before. Okay, I want to point that out to you. Whenever you are looking at any sort of scan, graph, trace, anything that has an axis, these things mean nothing if the axis is not labeled. So you have to always look at the labeling of the axis. So here we have the membrane potential on the y-axis. Now over here we have the distance. Okay. Distance within a few millimeters. Okay. So the distance, you can imagine, this is where the actual depolarization begins. This is where the rock is hitting the water. This is where the ligand or neurotransmitter is binding to the ion channel. We see the biggest response closest to that point, and as we move a distance away, we have less and less of response until the point where there is no response whatsoever. Okay. So that's just another way of looking at graded potentials, right? So these are only good over short distances. So we're going to discuss action potentials at a bit more in depth now than we did in Chapter 9. But just to review, when we talk about action potentials, we have a brief reversal of membrane potential, right? So going from resting membrane potential, where we have an approximate difference of 70 millivolts, so we measure the inside as being minus 70 millivolts compared to the outside. This is something that's unique to muscle cells and neurons, which we had said are the excitable cells. We don't see this change. All cells have resting membrane potential, but only muscle cells and neurons have the ability to change it and use it as a signal. Okay. Now, the key thing with action potentials when compared to something like graded potentials is that action potentials do not decrease strength over distance. Okay, graded potentials are strongest at the point where the stimulus occurs and get progressively weaker. However, action potentials don't do that. If an action potential got weaker the further that it went from the source of stimulus, then they wouldn't function for what they have to do. Action potentials have to be able to send signals along neurons that can have axons a meter in length. Okay. So let's start off. We've talked about resting membrane potential. This has to do with the relative numbers of sodium and potassium ions, so we have more sodium ions outside of the cell than inside of the cell, creating a concentration gradient and wanting sodium to move in. We have more potassium inside the cell than outside of the cell, so there is a concentration gradient so that potassium wants to move out. This is established in part by the sodium-potassium pump, which is actively pumping sodium out, actively pumping potassium in. Okay, and then the actual difference there is some degree of passive or leaky channels where potassium is leaking out and sodium is leaking in. At that resting state, we have these voltage-gated channels, and we're going to see the highest concentration of these voltage-gated channels where action potential begins, right? Where do we see action potential beginning in a neuron? Yes, the axon hillock. Very good. Okay. So what we're going to see is throughout the dendrites, throughout the cell body, there are some voltage-gated channels, but for the most part, what we see there are mechanically gated or ligand-gated channels. In the axon hillock is where we have the majority of these voltage-gated channels. Right? Now at resting membrane potential, we have sodium channels and we have potassium channels, and they're both closed. So at approximately minus 70 millivolts resting membrane potential, both of these channels are closed. Okay. There is that little bit of leakage. Okay. Now the sodium channels are pretty cool. The sodium channels have two door systems on them. Okay. They have two gates. The sodium channel has what's called an activation gate, and an activation gate is a gate that opens when the correct voltage is hit. There's also on the sodium channel something called an inactivation gate. Okay. The inactivation gate is a way to close the flow of ions through, or stop the flow of ions through, without closing the activation gates. So the activation gate and the inactivation gate are going to be sensitive to two different voltages. Okay, so we're going to have a way to open it and a way to close it at two different voltages. Now the potassium channel has only activation gates. Now the first part of action potential is the depolarization phase. Okay. What happens at the depolarization phase is that we're going to see the sodium activation gates opening, so the sodium channel's open, and sodium will start rushing down its electrochemical gradient and rushing into the cell. As it rushes into the cell, we are moving positive charges from the outside to the inside, and we are depolarizing. We are going to change that. Now, the way that this occurs is we have to have the correct voltage to open this activation gate to begin with, and we call that a threshold. Okay. The threshold, again, is variable. I like to use the number minus 55 millivolts. Your book also uses the range to minus 50. If you remember minus 55 millivolts as threshold, you will be safe in terms of testing and so on and so forth. Okay. Now, once that threshold is reached, we would see these voltage gated channels opening. What do you think provides the small depolarization to go from minus 70 to minus 55? What can bring us to threshold? Where does that depolarization come from? Yes, the graded potentials. This is why we had to talk about the graded potentials. Okay. The graded potentials that we see occurring at those dendrites, they happen for a reason. Okay. They are the initial depolarization. If there is enough of them, and by enough I mean that they can take at the axon hillock, they can take the resting membrane potential of minus 70 millivolts, and they can bring it up to minus 55 and hit threshold. Okay. The way this occurs is through spatial summation, temporal summation. So each of those graded potentials can bring in some amount of depolarization. If there is enough depolarization that we hit threshold, then we open these voltage-gated sodium channels. Okay. That's going to kick off the true action potential, and this is where we're going to get that self-generation. What happens here is when the first, if we imagine these just as a single line of channels, it's much more complicated than that. Okay. Imagine these as a single line of channels. Right. That very first channel needs all of those graded potentials to add up to hit threshold, okay, to bring it up to minus 55 so that that activation gate can open. Once that activation gate opens, it's going to cause a depolarization. That depolarization is going to be enough to hit threshold for the next channel, which is going to open and have a depolarization that's going to be enough for the next channel. That's going to open and have a depolarization that's going to be enough for the next channel, and the next channel, and the next channel. That's where it becomes self-generating. That's where it becomes self-propagating. Okay. It's like knocking over dominoes. Once you get that first one knocked over, the energy of the first knocks over the second knocks over the third knocks over the fourth. And it's going to go the entire way down the axon in a depolarization phase. Does that make sense? Yeah? Okay, we'll see. We'll also see this in an animation that might help. Now, the second part of this is the repolarization phase. So if we imagine staying still and we're watching just this one channel, this one sodium channel, okay, sodium is going to continue to come in, right? And we're going to see depolarization. It's going to be threshold, minus 55. It's going to go up, up. We're going to get the reversal of membrane polarity. We're going to go up to plus 30. Okay? More and more plus are coming in, coming in, coming in. And then at plus 30, when we have that reversal, something else happens. And this is where we're going to enter the repolarization phase. And you can really think of two things happening simultaneously here. The first, that inactivation gate of sodium is going to swing into place and it's going to shut. Right? So now sodium can't move anymore. So we have no more positive ions coming in. We also then have the activation gate of the potassium channel opening. So now these positive potassium ions are going to start rushing out. When these start going out, we're now taking positive charge from the inside and moving it to the outside, so we're going to start repolarizing. We're going to start moving from plus 30. We're going to get less and less and less positive inside until we flip it and go back to rest, right? Now what actually happens here is these potassium channels, they stay open a little bit too long, okay? And it's a good thing that they do. They stay open a little bit too long, so a bit more potassium comes out than really has to, and that actually goes into hyperpolarization. In the hyperpolarization, then the potassium gates stay open, an excessive amount of potassium comes out, right? Efflux means exiting. And this is where we're going to have the neuron being insensitive to stimulus. And that's going to be very important because that's what's going to keep these signals in the action potential in the axon moving in only one direction. So if we think about this, we have our depolarization, repolarization, hyperpolarization. Okay. At the end of this, we have to reset everything so the next action potential can come through. Okay. Again, if you were thinking of lining up dominoes and knocking them down, before you can knock them down again, you have to reset them. Right? And in terms of resetting them, we need to do two things to reset them. Okay. The actual action potential itself resets the potential difference. Okay. But what happens then is if we've had all the sodium rush in and all this potassium rush out, are we back at rest? Are we what we want to look like at rest? Is that what we need at resting potential? No. Okay. We need lots of sodium outside and lots of potassium inside. So in comes the sodium-potassium pump. Okay. Now the sodium-potassium pump is always working kind of in the background, but once those channels close, this is where we can restore the electrical conditions and the ionic redistribution. Okay, because the sodium-potassium pump is going to start pumping all that sodium out, pumping the potassium back in to get everything reset up so the next action potential can come through. If this didn't happen, you'd send an action potential down a neuron, and that neuron would then be broken, right? You wouldn't be able to use it anymore. Now, for a lot of you, this is easier to see graphically. Okay, so what we've just talked about, now we're going to look at it. And we've seen this trace before. We're going to talk about the trace a little bit more. Okay. Again, this is a trace, so whenever you look at a trace, a graph, a chart, make sure you know what you're looking at before you start looking at the lines. So down over here, we have time in milliseconds, so 1/1000th of a second. This is membrane potential in millivolts, and then this is relative membrane permeability. So the red line, the thick red line, is read off of this y-axis. The orange and green lines are read off of this axis. Okay. So this is telling us if we took a little tiny voltmeter and somehow were able to magically look at any point on this axon and see what happens as an action potential passes through any point on the neuron, this is basically what we're going to see. Here, number one, this is resting state, minus 70 millivolts. Where do we have a higher concentration of sodium? On the outside. Where do we have a higher concentration of potassium? On the inside, right. Okay. Here we have this initial increase. Okay. This initial increase up to minus 55, which is threshold. Where is this initial increase or this initial depolarization? Where is that coming from? Graded potentials, right? That are coming from the dendrites of the cell body, usually. Once we hit, my mouse keeps disappearing. Once we hit minus 55 millivolts, right, that's threshold. That opens those voltage-gated channels. When those voltage-gated channels open, okay, we see this very rapid depolarization. Right? You can actually see the channels open with this orange line. This orange line is the permeability to sodium, and that really means number of channels open. So once you hit minus 55, you see, boom, huge amount of channels opening. That huge amount of channels leads to this very rapid depolarization, up until this point right here of plus 30. Now at plus 30, we've had lots of sodium go in. So much sodium has gone in that we've actually reversed the membrane polarity. Now the inside of the cell is relatively positive compared to the outside. And a couple things are going to happen. Where's my mouse going? First thing we're going to see is that suddenly there is going to be a shutting of those sodium channels, so we're going to see a rapid drop-off in permeability to sodium. Okay. That means the channels are shutting. The other thing that's going to happen, we're going to see a fairly slow and steady increase of potassium channels. Right. So now we've shut the sodium channels, we've opened the potassium channels, potassium starts flowing out. We're taking positive charge from the inside, moving to the outside. That is going to allow us to repolarize. So we go down from plus 30 to 0, down. Now because this permeability to potassium, or these number of potassium channels, stays open a little longer, it's a little slower to open, a little slower to close, that is going to let more potassium out than necessarily needs to come out, and that's going to lead to this part right here, number 4, which is hyperpolarization. Okay. The hyperpolarization just means that more potassium has come out than really needs to, but it's going to be very important in how neurons work. Okay. Then to get back over here to number 1 again, where you could imagine this looping around or continuing, that's where the sodium-potassium pump has to come into place to start pumping that sodium back out, pumping the potassium in to get us back to rest. Now when we look at that trace, what we're really looking at is what's going on at a single point on an axon over time. So what we need to try and then transition is, how does this make a signal move? If we imagine the axon is, a long tube, at some point we have to kick this whole thing off and have that initial depolarization. That starts off at the axon hillock. The depolarization that comes in from the graded potentials gets at the axon hillock. The axon hillock acts as the trigger zone. Once that kicks off the action potential, the way this actually moves along the axon is like this. Okay. You have an initial depolarization that's going to open the next channel. Okay. So a depolarization is going to be big enough, if it's big enough, is going to open the next channel in the line. So we have this movement of these positive ions towards where it's more negative. So it's actually going to lead to spread. So when these positive ions, these sodium ions, come in, they're attracted to where there's less, right? They want to spread out. It's diffusion. So they don't just come in and stop. They come in and spread out. And when they do that, when they come in and spread out, that causes enough depolarization to open the next channel. And then they're going to come in and spread out. Now the key thing here that's very different than in the muscle, action potential from that neuromuscular junction, it went in all directions. It spread out in all directions. This, we want it to just go in the forward direction. So when we are looking at our point, where we have our little voltmeter, okay, where the little triangle is, as that action potential passes through, we see our typical sodium rushing in, we get that depolarization. When we look at that, those positive ions that are coming in are only spreading in the forward direction. Okay. The reason that this happens, the reason we don't see the action potential going backwards, is notice behind the red area of action potential, we have this gray area called hyperpolarization. This hyperpolarization is going to be where you have... Are you taking a picture of the screen? Oh, you know these notes are all available on Blackboard, right? Oh, I can totally get them to you then. You don't have to take pictures of all of them. If you want, if you ever need to get the notes, let me know. I can get them to you. Okay. I was like, I've never noticed him doing that before. All right. I'm sorry. I got distracted for a second. So this hyperpolarization, right? Remember we said that more potassium rush out than really need to be. They make that membrane potential even more negative. What that does is that pushes the membrane potential even further away from threshold. So what that means is graded potentials are not going to be able to sum up and hit threshold because there's a much bigger distance. Once this initially the whole thing passes, okay, that's what's going to keep this moving in the forward direction. This positive here is not going to be able to reopen these channels because these channels over here, this area here, is in a state of hyperpolarization. It's in what we call a refractory state. That refractory state we can call a refractory period. Okay? And there's really two refractory periods. There's an absolute refractory period. The absolute refractory period is from when the sodium gate initially opens until the activation gate closes. This is pretty obvious. You can't open a door that's already open. Right? And it's one of these things where, like philosophers would have a field day with this. Like, how do you even know the door exists? But when you have a channel that's already open and already undergoing a depolarization, it can't experience another depolarization when a depolarization is already occurring, right? You cannot fall down again when you're in the middle of falling. So that's the absolute refractory period. Okay? No matter what the circumstances are, you're going to only have depolarization occurring once at a certain point while it's still going on. The second part that has to do with that hyperpolarization, and this is the relative refractory period. The relative refractory period is during that hyperpolarization. The difference between where the cell is, where the membrane potential is, and where threshold is now so great that it's really, really, really unlikely that you would be able to get another graded potential strong enough to start an action potential. I won't say that it's completely impossible, but because we have this elevated level of difference, right? This much bigger difference between threshold and the membrane potential. Now, this hyperpolarization can last a period of time, okay? And if you notice that it's not always the same. So if you have fairly weak signals, a fairly weak signal would have to wait until the hyperpolarization is completely done before it could send another signal. And that's going to make those signals space out. Okay? Whereas if you have some pretty strong signals, they may be able to send a signal pretty quickly as hyperpolarization is finishing, because they might be big enough to bump it up this much. Okay? Now that the absolute and relative refractory periods are what is going to keep action potential going in a single direction, right? Because the part of the axon behind the signal is in one of those periods. Okay? The other thing that these action potentials are going to have because of this refractory period and threshold is a few important traits that are action potential. Action potentials are all or none phenomena. Right? So either an action potential happens or it doesn't happen. Now I'm going to notice every single time that he's taking pictures. Action potentials are all or none phenomena. So what that means is they either happen or they don't happen. There's no like halfway between an action potential and an action potential not happening. Okay? When you have a stimulus that's below threshold, you just don't get an action potential. If you have anything that's equal to threshold or greater, then you get an action potential. Right? It's truly like it happens or it doesn't happen. Now that leads to a kind of problem here, in the sense of there has to be a way for neurons to tell the brain what is a weak signal versus what is a stronger signal. But they can't do it by changing the size of the action potential. So an action potential is always going to be minus 70 to minus 55 to plus 30 to hyperpolarization back to minus 70. Okay? You can't make it bigger. Okay? You can't increase the amplitude. When you talk about a graphical distribution, an amplitude is the height. So what we have for stimulus intensity then is you can't make the action potential bigger, but you can make it happen more frequently. And this is actually due to the fact that with a stronger stimulus, that stimulus doesn't have to wait as long for that hyperpolarization to end before it can send the next one. So here we have, again, a different kind of trace. This bottom part is, again, this is going to be time in milliseconds. It's the x-axis is the same for both of these. This is the voltage. this is going to be the membrane potential. So here what we're doing is we are inducing a signal using an electrical stimulation. So we're trying to stimulate at the axon hillock to say, what do we have to do to make an action potential occur? Right. If we have a stimulus that is below threshold, then we have no action potentials. Nothing happens. It's going to be below our threshold. So we don't open any voltage gated channels, no action potential. Right. Here we have two different stimuli. One would be a weaker, one would be a stronger stimulus, but they're both super threshold. They're both at or above threshold. So they're going to induce action potentials. So the way that this can be coded in your brain, so that the way that this can be interpreted, has to do with the intensity. So if you have a stimulus that is leading to the sodium channels opening, it's going to lead to an action potential. Now, what these straight up and down lines are, these are what we've been seeing before. They're just squished over time. So this is showing you an action potential. Depolarization up to plus 30, down, hyperpolarization back to minus 70, rest. Okay, we've just taken that, we've collapsed it over time. So when we have this kind of weak but above threshold stimulus, we have an action potential, and the frequency is like this. Right? So if you imagine 1, 2, 3, 4, and each of those beats is an action potential, completing its entire course. When we switch over and we make a much stronger stimulus, okay, we don't change the size of it, but we have a higher frequency. So now we have 1, 2, 3, 4, 5, 6, 7, 8. Okay. Action potential, action potential, action potential, action potential. That's how you can code for stimulus intensity. Right? So if you feel the difference between a weak stimulus and a strong stimulus, right, the way that's interpreted is not the size or the amplitude of the action potential, but the frequency of the action potential. That's really, really, really important. You can't stop an action potential, right? You can't start it and then stop it. It starts and it goes all the way through, and it's always the same size. So all you can do is send another one directly behind it, another one directly behind it, another one directly behind it. All right, so as this occurs, as we have these channels opening, you can imagine this being continuous, where all along the entire length of the axon, you are allowing this sodium channel to open, positive sodiums rush in, depolarization, then we have the repolarization, sodium channels close, potassium comes out. But what's going to happen then is that would take too long for it to actually be functional in an organism as complex as we are. Right. In order to increase the speed that this can happen, there's like two things that you can do. Right. One of these is to increase the diameter of the axon. Okay. If you increase the diameter of the axon, you're increasing the surface area. If you increase the surface area, you could put more channels in. If you can put more channels in, you can make this action potential move faster, because the depolarization can happen quicker. Right? If you only have one channel open, you have to wait until enough sodium goes in that one channel to make that depolarization. If we open it up and we make the surface area much bigger, and we have lots and lots of channels, we can have lots of sodium coming in, so that same amount of depolarization can happen much faster. You can see this if you think about your house, if you think about, and you maybe have never thought about this before, but if you compare an electrical cable for something like headphones, right, or a cell phone, right? They're very thin cables, right? There's not that much energy that goes through them, right? But if you have like an RV, and you drive an RV and you plug your RV in at the campground, those cables are really thick, right? If you have an outdoor electric weed whacker, lawnmower, leaf blower, whatever, you use those orange cords that are pretty thick, okay? The thicker the cable is, right, the more surface area, the more energy can pass through it faster. Same thing happens with axons. Okay? So there are some organisms that just increase the size of their axon, make it bigger and bigger and bigger to increase the speed. One of the ones that was first studied is in something called aplysia. Aplysia is a sea slug, and they actually have a neuron that you can see. It's about the thickness of a human hair. Giant squid, or not the giant squid, but really big squid axons, so the axons are big, not the squid are big. They have an axon that you can see. You can see it visually with your eyes, right? None of the axons in our bodies we can see, right? So why, in the case of humans, does it not work just to make the axon bigger? Why can't we just, to speed up transmission, why don't we just make all of our axons bigger? We don't have any more room, right? I don't have a lot of extra space floating around my body, right? We are packed pretty tightly. If you take a look at the human anatomy, our nerves, could you imagine if suddenly all of our nerves increased by 100%, okay? You'd just basically be a walking thing of, we'd look like a mop. We'd be just nerves everywhere, okay? So we have to do something else, and this is where we have that myelin sheath coming in, right? So we talked about the myelin sheath. We said that in the peripheral nervous system, it's created by what kind of cells? Schwann cells. In the central nervous system, what makes the myelin sheath? Central nervous system, myelin sheath is? Oh, yeah, oligodendrocyte, right? Oligodendrocytes. So, again, that's one of those things you probably will have to just rely on recognition for it. It's actually not that hard, oligodendrocyte. What that myelination does is it's going to increase the impulse speed, right? The way that it does that is through something called saltatory conduction. Saltatory conduction is when the action potential gets passed along not in a continuous fashion. It's more discontinuous. So that at the axon hillock, the action potential starts. And then where those positive ions move forward, now they're going to slide all the way forward to the next node. Right? I will tell you, this diagram makes it look like the red lines are somehow the charge is jumping outside. It's really not jumping outside. It's happening within the axon. So if we look at what's going here at this node of Ranvier, the action potential has arrived. The depolarization has opened these voltage-gated sodium channels. Depolarization occurs, and when that happens, because this is insulated, insulated means that you're not going to have any ions leaking to the outside, okay? Because it's covered. Remember, we're talking about a plasma membrane here. We're talking about ions coming in and out. But if we cover that, if we seal it with this myelin sheath, they can't leak out here. They can't go out here. They're going to slide or jump down to the next open spot or the next node. Okay? So we have action potential concentrated here, and then it jumps. So we get that occurring in a jumping fashion. The saltatory conduction comes, if any of you guys ever have watched Dora the Explorer, or any basic Spanish, the verb saltar means to jump. So salta, salta means jump, jump. So this is technically jumping conduction. Okay? And what we see is the action potential jumps from node to node to node. So what would happen is, if we kind of overview everything now using this diagram, okay, you'd have a signal that comes in from some other receptor cell or some other neuron, leads to graded potentials here in the dendrites and in the cell body, the receptive region. If those graded potentials are big enough to reach threshold, then here in the axon hillock, we'll have our first opening of voltage-gated channels. When those voltage-gated channels open, sodium comes rushing in. Okay. That sodium, as it rushes in, it can't do anything along this whole stretch of the axon because that's sealed with the myelin sheath. So it jumps or slides along the axon till the next node. Now here we have plasma membrane exposed to the outside. We're going to have channels. So this depolarization that started here in the axon hillock is going to open sodium-gated channels here. And that's going to lead to a depolarization, which is going to jump and open voltage-gated channels here. And it's going to jump and open voltage-gated channels here. And it's going to jump and open voltage-gated channels here. And the depolarization is going to jump and open voltage-gated channels here. And that's going to move it all along till we get to the end, which is not shown on this diagram, where the action potential winds up at the axon terminals. Now, this can really increase the speed of conduction. So if you imagine going from point A to point B in baby steps, okay, it's going to take some time. Versus if you try and go from point A to point B with giant steps, okay, you can go much faster. So that's how it's actually speeding it up. These charges, this depolarization moves really quickly. But now we're taking that very quick and we're making it even faster. And that's what's going to be necessary because we have neurons that run from our brain to the tip of our finger, from our brain to the tip of our toe, right? Some of these neurons run really big distances. Now, we won't necessarily see myelination in areas of the brain where we have really tiny short neurons because they don't need it. Because they're so short, they don't need to be myelinated, right? So we're going to see that some of them are myelinated, some aren't. And that's going to be that difference between gray and white matter, which we'll see in chapter 12. Now, you may have heard of multiple sclerosis. Unfortunately, is a fairly common disorder. It affects, and it usually starts affecting people in their college years. It leads to a whole bunch of different symptoms, and it's more of a syndrome in the sense that there's a constellation of different things that can happen. What this is due to is the myelin sheaths in the central nervous system stop functioning. They basically become hard and sclerotic, almost like scar tissue. And that's going to lead to those nerve fibers being severed. So you're actually cutting off or short-circuiting some of the different circuits that control the nerve impulses. Now, multiple sclerosis is autoimmune. It's what's considered insidious, so we don't really know what starts it yet. It has varying degrees of morbidity and mortality in the sense of I have a friend whose mom has had multiple sclerosis for the entirety of her life. My friend is a few years younger than me, and ever since I've known her, her mom developed multiple sclerosis not long after she had her daughter. So, and she has some times where she's fine and sometimes where she's not, right? And it kind of comes and goes. So this is just one example of how diseases of the myelin sheath do lead to very serious consequences. Okay, you do not want to try and have neurons that are functioning that don't have the correct myelination.